Assalamualaikum and good morning. Today, our group will present about challenges and future directions in aquaculture vaccinations by Group 5. Before we start our presentation, I want to introduce my group member. We have 6 students in our group, and number 1 is Siti Nordana binti Isnil, number 2 is me, Elena binti Janet Lim, number 3 is Christina Libya Suntai anak Imai, number 4 is Maswe Martin, number 5 is Onicha Avi, and the last one is Daniel Phillips.
Hello, my name is Maswe Martin, SQ2160318, and I will be giving the introduction to the aquaculture and role of vaccine. Aquaculture is the act of farming of aquatic organisms, including fish, mollusks, crustaceans, and aquatic plants. Farming includes the implication of some way of intervention in the process of rearing in order to enhance production. This may include regular stocking, feeding, protection from predators, and management. Aquaculture are deemed important for the future of sustainable food security as it plays an important role in the global food system by providing high quality and affordable aquatic food for human consumption. According to the Food and Agriculture Organization of the United Nations, aquaculture is already the main source of aquatic food globally and expected to continue to be a major source of additional aquatic food production as the world's human population continues. The types of aquaculture are divided into 3 parts, based on the habitat of the cultured species. First, marine aquaculture, for example grouper, freshwater aquaculture, tilapia, and brackish aquaculture, for example salmon. A vaccine is defined as a biologically based preparation that is developed to improve the immunity towards a specific disease or a group of diseases. Vaccines are considered as biological agents that elicit an immune response to a particular anti-agent obtained from a disease-causing infectious pathogen, according to Chokor and Turchik 2014. It has been estimated that as much as 10% of all aquatic animals are lost because of infectious disease, leading to a $10 billion in losses annually on a global scale. Hence, the important role of vaccine. Preventing diseases such as stemming from various bacterial, viral, and parasitic diseases that, if not prevented, would have devastated an entire stock. The use of vaccine also led to helping creating a more healthy fish, promoting growth. This showed that vaccine plays an important role in aquaculture and ensuring a long-term sustainability and reducing the loss caused by infectious disease, and thus ensuring a profitable aquaculture industry by mitigating the infectious disease risk and in addition to promoting the risk of aquaculture fish. That's all from me. Thank you very much.
Hello, my name is Nanda Philip Juluting, metric number BS2216051. So today I'm going to present about the state of aquaculture vaccination. So currently, it is the introduction of modern medicine to our industry to improve the livelihood of our species. And this is reflecting on our technology that we have. This includes a variety of vaccine that we can choose from to administer to our species. It is enhancing the performance of our vaccines by improving its content and the stability of it. Not to mention, this also promotes species-specific vaccines, making it more efficient and more species-centered. Next is the type of vaccination. It is separated due to its different composition. Firstly, we have the DNA vaccines, where the vaccine only includes a particular pathogens and antigens like protein or polysaccharides. It also only needs a few times of injection, but it is very long-lasting and very effective. Next is the subunit vaccination, where it uses plasmid DNA, and after injection it will induce the antigen in the body to produce cell in the body and improve the immunity. The DNA also produces cellular and humoral immunity, not to mention it is very stable. Next, we have the vectored vaccines that contains viral and vectors and very long lasting. Lastly, we have the authenticated vaccine. It contains a weakened live pathogen. It is strong and very long lasting, but this kind of vaccine can backfire since the pathogen can regain their strength and infect the host. Next, we have the delivery method of the vaccines. We have 4 methods of delivery. The first one is injection. This type of delivery is very rudimentary, but it is also very precise and very effective. But can cause stress for the fish. Next is immersion, where the fish is immersed in the solution of the vaccine. It is very suitable for a large amount of fishes, but requires high amount of the vaccine. Next, we have the oral administration, where the vaccine is orally administered to the fish via feed. It is very easy to feed them, and but the quality of the vaccine can degrade since it can cause digestion problems in the fish. And lastly, we have the spray method where this vaccine is sprayed all over the fish. It is very effective for small fishes, but it does not have a equal distribution. And that is all from me. Thank you very much. I will next pass to the next person.
Assalamualaikum and good morning. My name is Elena binti Jinut Lim, BS221605 and 08. Today I'm going to presentation about challenges in aquaculture vaccination. The challenges in aquaculture vaccinations, the number one is fish species and disease. Fish species and disease. The diversity of fish species itself was a challenge in vaccine development as they still do not fully understand the fish immune system and each fish species requirement, reagents or primer to elucidate host pathogen interaction. In addition, although injection is commonly used for Atlantic salmon, this may not be viable for some species, for example, tilapia. The challenges in aquaculture vaccination. Number two is challenges model. Challenges model. Testing the efficacy of vaccine requires standard in vivo disease challenges models that closely mimic the natural exposure route to the pathogen. Although more difficult to control and standardize, that injection challenge mayhood bed and cohabitation challenges best fulfill the requirement of natural exposure. Pathogen load and immunological market of protections can be used as proxies to test the efficacy of vaccine. If not experimental disease challenges mayhood is available or may not be reproducible. Field trials can be performed in certain circumstances. The point 3 is T-cell response. T-cell response is efficient antigen recognitions and representations are required for effective vaccination. Vaccine antigen are usually presented through MHC11 molecules to T cell, while DNA vaccination is mediated through the MHC1 route. Although it should be noted that both responses can be triggered simultaneously with one prevailing as the immune response develops. The challenges in aquaculture vaccination, the last one is B cell response. B cell response, with regards to B cell response, correlations with antibody productions does not necessarily infer a protective response. Passive immunizations can be used to demonstrate the protective effect of antibody. In fish, in contrast to mammals, there appear to be no increase in antibody level following booster vaccinations, and it is true thought that more cells are actually produced during the memory response rather than any increase in antibody level. That is for me, so I'm going to pass the next presentation. Thank you.
In my part, I will present about innovation in future direction. For innovation in future direction, they have two advantage vaccine technology and improved delivery system. For innovation in vaccine technology, they have four. One is recombinant vaccine, two DNA and RNA vaccine, number three nanoparticles-based vaccine, and number four subunit vaccine. For recombinant vaccine, these vaccines are genetically engineering an organism to produce specific antigen that elicit an immune response. They offer scientificity and safety, reducing the risk of introducing live pathogen. DNA and RNA vaccine. This vaccine include the injection of genetic material encoding antigen which host cell then express triggering and immune response. They can be rapidly designed and produced, offering a quick response to emerging disease. Number three, nanoparticle based vaccine. Utilized nanoparticles to deliver antigen can enhance stability and target delivery, improving immune response. This can also help protect the antigen from degradation in aquatic environment. And for the last, subunit vaccine. This vaccine use specific protein or peptide from the pathogen, reducing the risk of adverse reaction and improving safety profile. For improved delivery system, there are four. Number one is oral vaccination. Orally in non-invasive and less stressful of feed. Innovation and escapulation technique ensure the machine survives the disease threat and is also very effective. Number two, immersion and bed vaccination. Fish are immersed in a vaccine solution, allowing them to absorb the vaccine through their gear and skin. This method is less labor intensive but require larger quantities of vaccine. Number three, injection vaccination. Although labor intensive, injection ensure precise dosing and is effective for value or high-risk population. Advanced is automated injection system are recording labor costs and improving efficiency. And for the last is adjustment and delivery enhancer. New adjustments and immunostimulate can enhance the immune response, allowing for lower dose and potential broader protection. Delivery essentials such as mucoadhesive substance improve the uptake of oral and immersion vaccine.
I am Monica Matthews, SQ21603. In my part, I'm gonna talk about our case study, which is progress, challenges, and opportunities in fish vaccine development by Alexandra Adams, 2019. The purpose of vaccination is to prevent disease breakout, especially in aquaculture field. This case study was mainly focused in the vaccination used to prevent disease in finfish aquaculture, which basically for this case study is mainly focused on Atlantic salmon, which is Salmon salar, also known. While in a limited capacity, based on the current study, the state of the vaccination in aquaculture vaccine now has been commercially available for use in variety of finfish. A challenge do, however, exist as the majority of commercial vaccines are kill whole cell pathogen preparation administered by intraperitoneal injection. The case study also highlights the potential opportunities for the future direction regarding the vaccination in aquaculture. In the next slide shows the challenge and future directions of this case study, which is for the challenge. The most crucial step in developing an effective vaccine is identification of potentially protective antigen and confirming their protective response is in the host species against the pathogen of interest. This approach can depend on pathogen type, fish species, administration method, availability of reagent, and whether a challenge model has been developed to efficacy test the vaccine candidate. Identifying protective antigen is not easy and requires a variety of approach. As for future direction, the genomes of a variety of fish species are now available. In addition, with the reduction in cost of technologies such as the whole genome sequencing, the genome for specific pathogens are regularly reported. This enabling targeted vaccine designed for heterogeneous species. For example, the NGO, or should I say the non-government organization, with combination of the other co-authors, characterize over 300 Flavobacterium cycrophyllum species, which is mainly from the UK, and this produces an effective trivalent whole cell vaccine. That's all from me. Thank you.
Hi, I am Zaitin Ardana Bidesner, BS2216051. For the conclusion, aquaculture vaccination is essential for fish health and productivity, utilizing DNA subunit vector and live attenuated vaccines. Key challenges include developing species-specific vaccine, creating accurate challenge models, and managing environmental and regulatory issues. Future direction focus on advanced vaccine technologies like nanoparticles and mRNA vaccines, improve delivery system and sustainable practice. Continued research and collaboration are vital for overcoming these challenges and ensuring effective disease management in aquaculture. That's all. Thank you.